So this topic of multi-voxel pattern analysis is one that you're going to hear quite a lot about in this course, especially this year. We really have a great lineup of speakers for you. And so I'm just going to be spending about 90 minutes now to set the stage for what you're going to hear next week. You have Jack Gallant from UC Berkeley, who's been a leading force in the development of models that are theoretically motivated based on our understanding of information processing, for example, in visual cortex and now in the semantic system, and ways of measuring tuning functions in individual voxels that can allow you to make predictions about any possible visual image a person might be viewing or imagining, and now semantic content that they're listening to in a story or thinking about. So he'll tell you all about that. I'm not going to try to preempt his content by going into some of his work, although it's been extremely influential, and I often do speak about it. But we have him, so you'll hear from the source. We also have Jim Haxby, who is widely credited as being the founder of this whole business of classifying patterns of bold signal and trying to infer stimulus-specific representations or mental states. So he'll be giving a couple of talks on Thursday about that. And we also have one of his postdocs, Dr. Swaroop Guntupalli, who is going to be giving you an interactive lab tutorial on the Pi MVPA toolbox and showing you how to use some of those tools, maybe the hyperalignment. I'm not sure what specifically he'll focus on, but he's going to be leading that workshop next Thursday afternoon. And then you also have Pamela Douglas, another course faculty member here from UCLA, who will be giving you a lecture and a little, I think, a brief interactive demo of some other topics that I'm not going to touch on today about how to interpret MVPA data, particularly weight maps that come out of your classifier, and evaluating those in a way that doesn't sort of over-interpret them or misinterpret them. And today you have me, obviously, giving an overview of the field. So first, just by show of hands, wanted to see how many of you have done some form of pattern classification or representational similarity analysis on bold data. Okay, just a few of you. How many have tried to classify other types of data, just not fMRI? It could be other MRI data or behavioral, other biological factors. So some of you have some exposure to this. So the key insight, just to sort of open things up, is that there's a lot of information in these whole brain fMRI scans that we get. Even if you didn't scan the whole brain, you just scan one region of interest, you're getting signals from many different voxels, and those voxels are sampling from hundreds of thousands or millions of neurons, depending on how big they are. And so there's just huge multidimensional informational content in these images we collect, and we collect many of them over time. You might have the subject in the scanner for an hour or more, experiencing different stimuli, making different kinds of decisions, having different thoughts. And usually we just use statistics to look at the peak effects. We're interested in regions that show reliable differences in a statistical sense at the group level across subjects between two different conditions of interest, and we create these maps. Or you might want to get sort of closer to the data and look at effects in individual subjects. But we're still just looking at the peak effects, areas that show the biggest bold signal in one condition relative to some other conditions. And so in that sense, we're looking at the tip of the iceberg. And I think if you're sort of up on the literature over the past decade, you'll realize that the field has come to the conclusion that there's a lot more going on in these images that we could be taking advantage of if we sort of look below the surface. So underneath here, the ocean, the iceberg has much more structure. And when we only impose these thresholds and look for peak effects of mean signal, we might be missing a lot of other content that could be extracted from the images. So even just looking at the same map unthresholded, you can see that although these two regions that survive the threshold on the left were the peak effects in this particular contrast, the activity of the other regions isn't kind of equally low. It's this landscape. Some regions just missed the threshold, others have very low activity, and some might even have negatively signed activity, and that could be interesting as well. And so this landscape of activity can convey a lot of information if we use the right approach to sort of quantify and parameterize that and try to make conclusions about what it's telling us. So the take-home message just from the very beginning is that there's more than meets the eye when you look at these maps, and that's really an insight that Jim Haxby gave us. So trying to classify visual representations in inferior temporal cortex as people look at different kinds of stimuli. He noticed in these maps, I mean, up until this point in the field, people were interested in identifying modules or stimulus selective areas like the FFA and the PPA and the extrastriate body area, and areas that responded really strongly to one class of stimuli. But there are thousands or tens of thousands of different kinds of stimuli that one can experience, and it would be strange to think the brain was organized in such a way that you could find clusters for categories like boots and chairs and laptops and microphones and everything else. So some categories have this privileged status like faces, but what he found is that even for faces, there's this distributed landscape of activity, a pattern. And so yes, the fusiform cortex tends to show a peak effect, but the pattern itself, even if you remove the fusiform cortex from the map, the pattern itself might be something that's really unique to when you're viewing faces, and that's sufficiently different from when you're viewing boots or chairs and so forth. And so just looking at this pattern, he found that you could classify very reliably what class of stimulus the subject was viewing. And I'll let him sort of tell you the rest, although I think he'll sort of pick up with his latest work, because he's done a lot since that seminal science paper 15 years ago. So one reason to sort of pursue this endeavor is that sometimes an area of the brain will show equivalent activity across two conditions. For example, if you look in the auditory cortex and present a subject with syllables, rahs and lahs, you might not find the rah area or the lah area, just like you might not find sort of a chair area versus a table area if you looked in inferior temporal cortex. These areas might engage neural ensembles kind of broadly, and you'll get some bold signal, but it won't necessarily differ significantly in any cluster of voxels. And yet the pattern of activity, the local spatial pattern of bold within these areas could differ, and it could differ because it just so happens that neural selectivity profiles in neural ensembles that fall in one voxel are a little bit different than the next voxel over. So each voxel may not have sort of a significant preference for one condition than another, but it might have a slight preference, and if we look at the whole pattern, you might see, well, the pattern changes when someone listens to a rah and lah, the auditory cortex would differ. And then you could look at interesting questions. I mean, in this example from Rajiv Razada, he went on to look at how Japanese speakers and English speakers differ in their processing of the syllables, and this would only be possible with an analysis of distributed spatial representations of bold rather than peaks and valleys alone. Okay, so here's just sort of another example of how this kind of analysis works schematically. So you present different faces and different buildings in a task, for example, and you get these whole brain patterns for each stimulus. This could be individual trials, or you could average across several different examples of seeing that same stimulus. And you see this landscape of activity. So just a distributed pattern. There are some areas that peak more for faces that are just visually apparent in these upper two maps, and some areas that seem more responsive to scenes that are readily apparent. But if we use the whole activation pattern, we can try to compare them. And this is what Haxby did, essentially looking to see, well, if we compare this map to this map, it's a high correlation, and this map to this map. We're just looking within a small patch of cortex here. There's a low correlation. And so you might conclude, if you were looking at a map and you didn't know which condition it was, well, it's more similar to this one, the face one, than it is to this one, the scene one, or this one. So that's sort of the game you can play, and you can do it with just correlations. This is something called representational similarity analysis. I'll try to talk a little more about that at the end if there's time, but I'm going to talk mostly about multi-voxel pattern classification, where we're trying to train a classifier and then test it, and you end up with the decoding accuracy. But in representational similarity analysis, which is really how the field started in 2001, and then it kind of died off for a while, and now it's making a huge resurgence, you're just looking at the mean correlation of spatial patterns of activity across different trial types or conditions. And so here it's plotted with dissimilarity, so pairs here with low similarity have blue colors. They have high, so they have low dissimilarity. So they're in blue, and high dissimilarity is in the warm colors. So faces versus houses have high dissimilarity. The patterns are very different. And you could do this for any class of stimuli and try to see how does this brain region, does it care about the distinction between these trial types, or does it basically process them similarly? And then you look at another region. One region might care a lot about faces versus scenes, and another region might not differentiate those. And you can start to ask really interesting questions about different stimulus classes or mental states. Okay, this was just one. I thought I took this slide out, but one example where you have a hypothesis about, in this case, learning. Subjects are encountering faces during a scanning session, and some of the faces they'll encounter multiple times. Actually, each face here they get to see 4 times. This is a paper that was in Science a few years back. And just when you look at the spatial pattern of bold activity in a region, for each face, you get this distributed pattern here. This is just a schematic. But if you compare it from each face that you encounter, and you look at the faces that you later, after the scanner, go on to remember versus the faces that you go on to forget, the ones that you remember tend to have higher neural pattern similarity, that each time you encounter that face across the 4 different exposures, the more similar your brain activity is to the previous encounters, the better it is. You're sort of sharpening your representation or maybe reactivating it. Whereas if you process it in a very dissimilar way each time, in this particular study, they found that you tended to forget it. So that was just one example of how this can be applied, but you've probably seen many others in the literature. So going from sort of this representational similarity or correlation-based approach to a classification-based approach didn't take the field very long. So 2 years after the Haxby paper came out in Science, David Cox and Robert Savoy published what I think is the first paper trying to use a machine learning approach applying to bold patterns and just taking the Haxby data a step further. So they used the same kind of task where subjects viewed different classes of visual stimuli, and they extracted signal from regions in visual cortex, visual association cortex. So this is just a 200 voxel region of interest. Each point here is 1 voxel, and the voxel, its bold signal, varies across the region. So this might look like a time course, but there's no time here. This is just each voxel's response to these different categories: baskets, butterflies, cows, gnomes, and so forth. And just sort of looking at these patterns, you can't necessarily tell that they're very distinctive. And in fact, if you look at the mean signal of this particular region across all these classes, it's indistinguishable. So if we just overlay all of those, the mean sort of MRI signal for baskets and butterflies, there's no difference. And yet, when you try to use this classification approach, where you train a classifier on some set of your trials, it could be, in this case, I think they divided it up into blocks, but later people have, very, very quickly after, people went on to doing single trials or events. And you get these patterns for each trial, and you say, well, this is going to be my training set. I'm going to train my classifier model to learn sort of what's common about this class of stimuli, the baskets. And so we'll be exposed to baskets, and it will then be exposed to other categories, and we'll start to learn about what distinguishes these different categories. And then you just have some portion of your data that you hold out. It could be the odd runs versus the even runs. It could be, if you have 10 scanning runs, you could train on 9 runs and test on the 10th run. You could even just leave out 1 trial. And people do it in different ways. I'll get back to that a little bit later. But just leaving some portion of the data out, and then feeding in the pattern to your trained classifier and saying, well, given this pattern of activity, what do you think it's likely to be? And the classifier will make a guess. In this case, it's guessing basket, and you could see, is that right or wrong? If it gets it wrong, what category is it picking? Is it picking something that's visually similar to baskets? That could be interesting also. And so this is the general approach. And many of you are familiar with this, but for those who are less familiar, I just want one more sort of intuitive cartoon to convey the approach, and then I'll get more into the practical details that you need to think about if you go back to your lab and do this on your data. So you're going to acquire data while the subject is viewing 2 or more classes of stimuli, or could be performing different tasks or having different thoughts or decisions. But in this case, let's just do bottles and shoes. you get some pattern of activity throughout the brain. This example, there's just 6 voxels, but you could do this with all 200,000 voxels, or you could pick a region of interest and use 100 voxels, however you'd like. I will talk more about those choices later. And then for each trial, or each block, you just get some pattern of activity. So here's 3 trials where they're viewing bottles, and then 3 trials where they're viewing shoes, and so forth. And you can label these and tell your classifier sort of which is which. So these are the bottles, these are the shoes, bottles, shoes, and so forth. The classifier, and I'm leaving classifier right now as kind of a black box. There are many different kinds of algorithms that are effective at being presented with patterns of data and figuring out what the most sort of diagnostic differences are between those patterns, so that you can create some separating boundary in multidimensional space and be able to infer the class of new patterns. And so I will talk about the classification a little bit later, but for now it's just some algorithm that's getting patterns, being trained to identify the differences on the labeled patterns, and then it's going to make predictions on the held out set. And you have some feature space. It's easy to show a feature space if there were only 2 voxels, but if there's more than 2 voxels, the feature space is inherently multi-dimensional and can only be sort of conveniently visualized by a cartoon where you project it to some, project this multi-dimensional space onto 2 dimensions. So this feature space is basically, given all of the voxels sort of projected onto just these 2 dimensions, how would we separate the 2 classes? And that's what the classifier is learning. And in this example, it's doing pretty well. When you take these test patterns, this pattern it's getting correct, other patterns here would get correct, but this is just showing one example of a misclassification. So a bottle that was just on the threshold here of the classifier's decision boundary and would happen to get classified as a shoe. Okay, so abstractly sort of, or broadly, the goals of doing multi-voxel pattern analysis differ depending on what kind of questions you're interested in. So some researchers are interested in decoding. Decoding sometimes just for the sake of decoding. You're trying to see like how accurately can we tell if someone's lying or telling the truth. How accurately can we tell if they're seeing a stimulus that they recognize or seeing a stimulus that's novel, or experiencing a certain emotion. Or you could try to use those data you get about decoding accuracy and see how it explains individual differences in behavior or other psychological theories. But this is one approach. Another approach is trying to distinguish individuals from each other. So we have a lot of brains. People are doing tasks or even resting state or whatever you want them to do in the scanner. What's different about their brains? Can we tell which people are likely to develop Alzheimer's disease or not? Which people have ADHD, which schizophrenia versus bipolar disorder? People who are likely to have drug addiction versus those who don't. So this could be another strategy or motivation for using this kind of an approach. You might also be interested, and these bullet points are not mutually exclusive, but you might also be interested in characterizing the distributed cortical representations that are associated with specific cognitive processes. So if you're coming at it from sort of a cognitive neuroscience or cognitive psychology perspective, you can use the features that are being picked up by the classifier as a way to infer sort of what representations might be active in different areas of the brain, or what cognitive processes might be engaged. So this can help you understand sort of the mechanisms. Is this one of the topics we chose to debate? I forget whether there was something like this. Well, can you learn anything meaningful from multivariate classification? And I would argue that you can, although it is tricky. But there are many psychological theories where you posit that some region is or cognitive neuroscience theories that posit some region has containing a certain type of representation, and storing information in working memory, or maintaining your rule representation, or coding for reward value, and this could be one way to test that. You can also use it to test just purely psychological theories. So what, by measuring the engagement of mental states over time, you can see, for example, if, during a, well, let me see, there are a lot of different examples of this. I'm from the memory background, so like during a working memory task, like you might posit that there's sort of like active maintenance of the stimulus. That would be a psychological process, but it might have a neural correlate that you could, if you can continue to decode the stimulus that the subject is holding in memory throughout a period of time where they're just sort of waiting for some probe to occur, then that would suggest that active maintenance is happening. There are other examples where you can sort of see things about capacity limits and other kinds of psychological constructs. If maybe your theory has to do with people activating a certain emotion during a task, and you're inferring that that's happening in your psychological theory, and this could provide a way to test that if you could measure the pattern that's associated with that emotion versus other potential candidate emotions, and then read it out on individual trials and relate it to their behavior. This could be useful. Okay, so we're going to get now into the practical sort of nuts and bolts here, and this will be sort of a cooking show style demo. I'm not going to sort of lead a lab exercise and walk you through the individual steps, but I'll show you sort of quickly each of the stages that I think are important to think about as you're setting this up. And the field is changing so rapidly, it's such sort of a moving target that even since I prepared these slides a few years ago, there are other different techniques and newer toolboxes out there. But the toolbox that I've used the most in my own research on this is one that was developed at Princeton, the Princeton Multi-Voxel Pattern Analysis Toolbox. At the time I started using it, it was one of the major resources available, and there was a lot of development going on. But it's since, even though it's still supported and they occasionally make updates to it, it doesn't have an active development community. And the Pi MVPA toolbox, which you'll do a tutorial on next week, has become more dominant of a force, both because Python is as sort of an open source computing platform has become preferred over MATLAB by many labs that are doing this kind of work. And also there's just more active development happening on this team. So they use the interface that the Princeton folks came up with, and then they've added a lot of functionality to it. I just haven't made the switch yet in my lab. And then there's the pattern recognition tool for neuroimaging toolbox, or Pronto. I haven't played around with this. There are others also that I haven't sort of put screenshots in. Brain Voyager has its own package. I've not used this, but it's built into Brain Voyager and has kind of a GUI interface that might be useful for people who don't have a lot of programming experience. Okay, and just as a suggestion, I mean, this paper is already a few years old, but Francisco Pereira, who has been a former NITP lecturer and student, wrote this paper with Tom Mitchell, who's a real leader in this field at Carnegie Mellon, and Matt Botvinnik, who has also been working on it now. He just left Princeton to go take a very well-paid job at Google DeepMind. They wrote this tutorial, and there's a lot of useful material that's still relevant today, so I just point you to that piece as a good starting reference. So now I'm going to sort of walk you through some of the steps. So we have fMRI data, maybe you collected it for some other purpose, but your advisor tells you you should do MVPA on it, or you want to do that, or maybe you designed it specifically for this, but what do you want to do to your signal? And many of the things you want to do are the same things that you would do for univariate analyses or connectivity analyses, that you want to try to make the data as clean as possible. So removing signal artifacts, which is something we'll hopefully have more time to talk about in a separate presentation, is useful here, very useful, especially if you're looking at single trials. Detrending, so this is becoming less of a problem now that scanners have more stable gradient hardware. Our scanner here doesn't show a lot of drift within each run, where the mean signal level of a voxel will just sort of linearly decline or increase, but scanners used to have less stability. It's still useful, I think, to detrend, but this won't have as impactful of an effect these days. But high-pass filtering does, so there's a lot of low-frequency noise in bold data. Most of the noise is sort of at lower frequencies, and removing that with an appropriate filter is beneficial here, just like for univariate analysis. And then z-scoring the data from each run so that each voxel has a mean signal of 0 and a standard variance of 1 is typically done as well. So you're going to be looking at the patterns, but you don't want necessarily, if you have multiple runs of data, to have concatenation problems where one run had a mean MR signal of 872 and the next run it was like 950. And so at least equating the runs is what you're trying to accomplish here. Okay, next you want to parse your data into what I'm going to call examples. These are the individual patterns that you're going to train the classifier on, as well as some that you'll test the classifier on. And there are a lot of choices about how to do this. So for block designs, people will just typically either average all of the time points within a block. So if you have a 20-second block, you could just average those 20 seconds. You might want to do a little bit of time shifting to account for the hemodynamic delay. So you can start 4 seconds into the block and decide if you want to truncate at the end of the block or go a few seconds past the end. But that's one approach. Or you could model it with a regressor. You could have a sort of a boxcar regressor convolved with the HRF, run a GLM, get a single block beta for that block, and use that. And you'll get pretty good signal for block design. All of the initial work by Haxby and some of the earlier studies use block designs. And for some questions, that's adequate. But for many questions, you want event-related design, so I'll get to that in a moment. You can also treat each time point as an independent example. Some of the papers I've seen that do block designs, as well as even some of the tutorials, do this. You just sort of, every TR, you're just labeling it as, is this TR sort of condition A, condition A, condition A, condition A, and then the block ends, and you might have some ambiguous TRs that you throw out, and then you start calling them condition B, condition B, condition B. This will inflate the size of your training set and testing set, but it's a little bit artificial because those examples are non-independent. They're very correlated because of the sluggishness of the bold signal and the auto-correlation of our images over time. So for event-related designs, generally, people will take an approach like choosing a single post-stimulus time point or averaging several. I've found this approach to be pretty effective. So if you, especially for a widely spaced event-related design where you have some trial that occurs, they see a stimulus, make a decision, you wait 8 or 10 seconds, then the next trial occurs. You could just take the data from like 4 to 8 seconds post-trial onset, the peak bold response, and average that, or do a weighted average, or look at the difference from the start of the trial to the peak, and somehow just measure that effect in the raw data, and that could be your pattern. So you need long inter-trial intervals for this to work. So if your events are rapid and jittered, just averaging the data 4 to 8 seconds post-stimulus will be kind of sloppy. You might want to do something else, which I'll talk about later. And another important, just an important consideration is the effect of balancing the number of examples from your different classes. This is not always critical. Certain classifiers are more sensitive to these imbalances. And it's particularly important for how you're going to interpret the classification accuracy results you get. If you have 70% of your trials are one condition and 30% are another condition, and your classifier just develops a bias, it learns that, oh, I'll always guess condition A because that does pretty well. And then in the testing set, it's sort of always guessing condition A. Well, we'll get the 70% of your trials right and 30% wrong, and it will look like 70% accuracy, but really, if you did some D prime measure or corrected for the false alarms, your classifier hasn't learned anything. But if you balance your trials, so there's always 50% of class A and 50% of class B in the training set, or for multiple classes. Let's say if you have 5 classes and the class with the fewest trials is just 12 trials of each class, so then you can just randomly throw out trials from the other classes. So you have 12 trials of class A, 12 from class B, and so forth, and do that for each training set and each testing set. And then your classifier is going to be immune to these kind of biases because it will always have equal numbers of patterns. So I find that useful. It helps me sort of have confidence and sanity when looking at the results. And if you don't do that, you should just be really careful to look at your results and see how the classifier is doing for each of the classes and if it's been biased to guess one class more frequently than the others. Okay, so going back to the issue of sort of creating these examples. So you have a design, in this case, a slow event-related design where every 10 seconds the subject sees a face and makes some decision or tries to memorize it, whatever is going on in your task. And you're measuring bold responses. These are particularly clean-looking ones, so maybe from a visual region, that are responding to the face. And we don't actually get signal like this. This is like the hypothetical bold response to each face. Really, we just get sort of one time course, and we're trying to make sense of it. But the approach that I was talking about just a moment ago, where you would extract several post-stimulus TRs that you hypothesize would be of particular interest, would be like this. You would just take the third and fourth, in this example, a 2-second TR, you would take the third and fourth post-stimulus TR, and that would basically give you the signal from 4 to 6 seconds and 6 to 8 seconds post-phase onset, which is when you would expect the peak bold response. So just averaging that and calling that your pattern is one approach here. You can also run a new GLM that estimates a single parameter for each trial, like the beta series approach I talked about yesterday, or the newer take on it, this sort of Mumford et al. Least squares separate approach. An alternative strategy would be to, and this is useful, and I'm seeing it more and more, you just train and test separate classifiers using data from each post-stimulus TR. So what I mean by this is that you don't really know sort of where to expect the peak signal necessarily, or you're willing to let the data show you where it is. Or maybe you're even interested that the prefrontal cortex can decode something first and then parietal cortex decodes it later. And if you have a fast enough TR, you might see that the peak decoding in different regions differs. So what you do here is you just treat your first post-stimulus TR as your training pattern, and you train and test a classifier and you get a result. And because the bold response hasn't risen at all yet in those first 2 seconds, the classification result should be near chance level or very close to it. There's some of the early bold signal could be influencing it, but generally these are just data I computed on some a memory task in my lab. Should be close to chance at the beginning. And then once you wait sort of to the second TR, now 2 to 4 seconds in, there's a little bit of information. And then if you were to train and test classifier using only data from the third TR, it's doing the best. The fourth TR is doing pretty well, not as good as the third, and so forth. And so the classification accuracy follows the time course of the bold response. And this is what you'd expect. Sort of here's another example. I should have updated this paper. Came out, I think, in 2015. So you train and test separate classifiers using data from each post-stimulus TR, and you can compare different regions. You can see that this region of inferior frontal sulcus did really well. And the third, well, this is in seconds here. 3 seconds relative to stimulus onset. And other regions did well later. Here's just a few more examples of studies that I've been involved with that have used this approach, which just gives you classification accuracy, or in this case, it's just sort of evidence for reactivation of a particular mental state as a function of time. So it's just the classifier output at each post-stimulus TR. So any questions on anything I've talked about so far? Yeah.
Do you have a sense of,
I'll repeat the question.
do you have a sense of if you're doing slow event-rated design, how long like the minimum?
Okay, so the question is, for a slow event-related design, how long should the intertrial interval be so that I can be confident that I could just sort of chop up the data and pick individual TRs? I generally find that like if it's shorter than 7 seconds, it starts to get problematic for a fixed duration. Most slow event-related designs use either a fixed duration ITI or a little bit of jitter, but it's still like the minimum ITI is long, like 6 or 7 seconds or longer. If it's jittered such that some events only have like a 2-second or 4-second gap and others have a 10-second gap, you're going to get variable amounts of hemodynamic sort of residual contamination. Some trials will be really affected by the preceding trial and others won't. And so just simply sort of chopping out TRs and calling those your patterns will not give you as good of a result as doing some more thoughtful modeling of the hemodynamic relationships, such as using this least squared separate approach, which is starting to really catch on, I think. Good question, though. Yes? Okay, so the question is, can you train your classifier on a block design, test it on an event-related design? And the answer is absolutely yes, that's fine. You can train your classifier on a block design and send it to your colleague in another country, and they can apply it to their data. Once you have a trained classifier, as long as the other person can get their data into the same brain space, what you have is a set of weights for each voxel. The classifier has learned sort of certain voxels are diagnostic or predictive of one class and others of another class. So that classifier, it's sort of internal machinery, will be something you could apply to other things. Like I could have a classifier that detects whether or not someone's in pain that I've trained on a big data set, and then I could send that to other clinicians who might want to use it to see if someone's in pain. This kind of thing depends on the brain patterns being generalizable across subjects. The example I just gave, your example was a little more narrow, that you have a block design, like maybe for each subject they do a face for scene localizer, and I find the brain patterns that best diagnose whether they're looking at faces or scenes in this robust block design. And then I want to do a sort of memory task where they see words and they either have to remember if they were associated with a face or a scene. I want to see how much do they reactivate those face and scene specific visual representations. So I've trained my classifier in this block design, and then I can test it on individual trials for my event-related design, or I can even just test it on every single time point of my event-related design. Let's say my main task is 1,000 TRs long. I can just sort of get the classifier's evidence for each class at every single time point and then do event-related averaging on that. So you can apply your classifier to any brain pattern, and it's just a question of how you're going to quantify sort of the output. If you're going to, once you feed those patterns in to your classifier, depending on the way you've set up your classifier testing regime, you'll either get a probability for each class, so what's the probability that this trial is class A versus class B, if you sort of run it through some logistic function, that's usually how I do it, or you'll get an evidence score for each class, and then you can just either pick the one with the highest evidence, and that would be your classifier's guess, and compute the accuracy, or you could just, as in this example, you're just plotting those evidence scores. Yes?
Hi. I wonder, so in this particular slide, whether you use the time series data or the beta value of each post-stimulus TR?
This is just using the raw, raw meaning that it's not really raw, we pre-processed it, but just using the actual data. So we know when the stimuli occurred. We just load the entire time course of the whole brain into memory, and then we can either do it for whole brain or focus on a region of interest. So let's say you do it for a region of interest that's just 100 voxels. So now you have 100 voxels times, let's say 1,000 time points. And I know the times that events occurred. And so every time an event occurred, I just take the first TR, the TR that happened locked to onset of the event. And I train and test a classifier using only those. And the classifier does very poorly and gets no information. That's my first point here. It's like a 0, or basically 50%. And then I do it again, but now I take the second post-stimulus TR, and it also doesn't do very well. And then I do the third one, and it gets better, and so forth. So the information emerges over time, but there's no beta modeling in anything I've shown so far. Did that answer the question? I'm not sure. Okay. You can pass the microphone back, I guess.
So for this purpose right here, so what about if you have confidence about the hemodynamic response function, then you weight according to that function, then take that value?
Yeah, so you can, instead of trying to estimate the classification accuracy at each sort of post-stimulus time point, we could try to derive a single number for each trial based on a weighted function. You could just take the SPM canonical HRF or the FSL sort of double gamma and just look at sort of the weights of it over time, sampling it at the same sampling interval of your TR. I've tried that approach. It's basically, it's sort of similar to taking the peak TRs, except you're not, in my case, I just equally weight these top 2 TRs, and this is just a toy example, and give everything else a weight of 0. But instead, I could give sort of this one a weight of 0, this one a weight of 0.15, this one a weight of like 0.39, this one a weight of 0.47, and whatever the weights end up being. And then you just get a weighted average. It's a good approach, and it becomes quite similar to fitting it with the HRF and getting a single trial beta. So those are 2 similar approaches. And you don't have to use a canonical HRF. You could use an HRF that's derived from motor cortex, or that's sort of estimated for your individual subjects in some unbiased way. Okay, good questions. This was just it's probably not worth getting. This is just another example of a paper where the timing of the classification ended up being really relevant. So without going into all the details of what they did, they were just interested in whether they could predict whether people were going to push a right button or a left button. And the subject, in this case, the subject could respond whenever they want, and they were interested in whether the brain activity could predict what response they were going to make before the subject even realized they were going to make the response. And the behavioral paradigm had some ways to sort of measure when they realized they were going to make the response, but you don't need to know about that to sort of appreciate the fact in these classification time courses that these frontal areas could decode which button they were going to press like 8 to 10 seconds before the subject was going to press it. So the bold activity patterns within these regions start to show predictive value. Now it's only about 55 to 60% accurate, so it's not perfect, but starting to predict what decision the person is going to make before they had this sort of psychological marker of their decision. So the button press is the actual decision. The subject give the experimenter some clue as to when they felt like they had cognitively arrived at the decision, but the fMRI data preceded that by quite a lot, whereas motor regions, the decoding happens as you'd expect with the standard sort of HRF. So this red line is when the person made their actual response, and then the bold response in this area sort of goes way up, and it's different if you make a right button press or a left button press, and you can decode very accurately which button they pushed after they pushed the button. That's not so surprising, but these areas could decode which button before they pushed it. And many subsequent studies have sort of played this game. It's controversial, sort of whether it undermines free will. That's more of a philosophical question, but showing that information can be predicted about upcoming decisions from areas of the brain like prefrontal cortex or sometimes even sensory regions. So this kind of strategy of training and testing your classifiers on individual time points can be useful for that as well. Okay, another question. You got the mic, good. Sorry.
So how would you know if that's an acceptable, so you mentioned that paper had something like 55%?
Yeah, so I think this is something that I have some slides on, but it still is something I would consider unresolved. In the field right now, you can still get papers published with really low accuracy if it's significantly different from chance. And if your classifier is well-trained and well-constructed and unbiased, theoretical chance, which would be 50% for 2 classes or 33% for 3 classes, you can just test against that. Sometimes people will shuffle their class labels, and if you shuffle them 100 times or 1,000 times, you'll get your empirical chance. And if your classifier was set up properly, that should be basically, if you did it enough time, it should be the same as the theoretical chance. So I don't always find, it takes a huge computational resource. I usually do it for a few of the classifications just to confirm that there was nothing biased in the code. But whether you test against the theoretical chance or the empirical chance, you're just usually doing a t-test. It's like a 1-sample t-test of is 62% accuracy as a mean different from 50%, and so each subject has an accuracy. One is 53, one's 57, one's 72, and there's variance, but if they all tend to be sort of positively deflected above chance, you'll get a really high t-score. The problem with that, and I think it is a big problem, is that really low classification accuracy is like 51.8%, could sometimes be significant if all of your subjects tend to have accuracies that are just north of 50% and very few of it south. You could get a really high t-value, and now you might say, well, we significantly can decode like what product the person's going to buy from activity in this region, and it is significant, but it means that 51.8% of the time you could guess which one they were going to pick, and the other half of the time, more or less, you couldn't do it. So knowing the accuracy is important. Sometimes just showing T-maps, which people can get away with in searchlight analyses, like which regions had significant decoding, and I'll come to searchlight analyses, can be misleading because areas that have high significance at the group level don't necessarily have effects that would be significant in any individual subject. In individual subjects, the ways of estimating chance are very well known. Like with 2 classes, you could use a binomial probability distribution. And if I had 100 testing examples that I was trying to guess, like how unlikely would it be for me to get 60 of them right by chance? If I flip a coin 100 times and just guess, like I should get 50%, but sometimes I'll get 60% right. I mean, if each of you did it and wrote down your score, there would be a distribution, and that distribution for a fair coin would follow the binomial distribution. And so it's easy to determine for an individual subject if 55% is better than you would get with a coin flip. But the problem is that at the group level, there's no sort of way to deal with that. So most people just do these t-tests, and they're much more forgiving. Okay, good question. So what about rapid event-related design? So I mentioned this a little bit. You could, there's a few different approaches. One approach is that you just have these different trial types, in this case 3 different classes of stimuli, so type A, B, and C. And they occur at different time points with some jitter and randomization. And then you build this model. So if you're going to do your univariate analysis, you just convolve each onset with the HRF, and you have a model for the stimulus A, condition A, model for condition B, and model for condition C. And then you could just look at each time point, sort of, this is the sum. This is just an illustration of what the sort of the summed model here would be. But this was the important part. You could look at each time point to see which class should be most active in the person's brain according to your model. So you would just look at your design matrix and see, like, is yellow the winning condition here? So I'll predict yellow, predict yellow, predict yellow, predict yellow, predict yellow, and now I'll start predicting condition B, condition B. And for some time points, you could just, if the difference between your different predictor variables is not big enough, you could call it ambiguous and throw these TRs out. So some people would just sort of choose individual TRs using their univariate model. At this point, you might sort of predict condition C, C, C. So, and when I say predict, I also mean this is how you would label it if it was in the training set. So you're labeling these patterns as paradigmatic of condition C. But then you get to this point where condition C is barely leading out against condition A, and you might have to score that one as ambiguous as well. So that's one approach, but it's not the dominant approach. Some people, although surprisingly not me, have used the Rissman et al. Approach. But this Mumford tweak, which I think, for reasons I didn't have time to explain yesterday, has some advantages in terms of how it is able to separate out variance that's coming from individual trials in a way that doesn't overfit the data quite as much. This is the estimated single trial activity for each trial. You sort of leave one trial or event out of the GLM. Well, you're not leaving it out, you give it its own regressor, you get the estimate for that, and then you sort of play that game again and again for every trial, and you save those beta maps and load them into your MATLAB program or your pattern analysis toolbox of choice and work with those. And that can be quite good for RSA or MVPA. Okay, so I explained this yesterday. And there's the reference. The first paper, they've done a few other subsequent papers on this that I urge you to take a look at. And I am going to try to contact Jeanette and see if I can share the code that she provided, or maybe she has newer code. Okay, so another approach is to run a standard univariate GLM to derive condition-specific betas for each run. So I've seen this also. If you have 10 runs or more, you could just sort of get an estimate for each condition for each run, and then train your classifier on data from 9 runs and leave out the 10th run. So you only have 9 training patterns of each class. It's not a lot, but those patterns will be better estimators of the condition-specific activity than single trial data would be. So you're sort of compromising the number of training and testing examples, but the training and testing examples you have will be hopefully well estimated. So if you have like 5 different conditions in each run, and they're all rapid and jittered, you're not going to have good success using beta series, or even the Mumford might not work so well. But you could get at least one pattern for each run and then work with those. Or you can even divide your runs into half. So the reason this works is that the way that the univariate GLM works is it's trying to sort of fit the data across all of the events of a given condition within the run. So sometimes condition A is close to condition B, and sometimes it's far apart, and sometimes there's another condition C that's intervening, and sometimes that's after. And so you have sort of this nice model, like you saw in this figure, fit the data that accommodates all instances of that condition across the run. And so you get a good estimate that deconvolves the condition-specific activity, taking into account the timing of the events that preceded and ensued. And this is a very profitable strategy for our univariate analysis, and if you have enough runs, you could try MVPA with that. Okay, so the problem is that if you have few runs, unless you're going to break your runs into sort of half and do pseudo runs, you don't have a lot of training data to give your classifier. So you could divide your data into mini runs. And as a important side note, a few years ago, Mark Coutange, I don't know if I'm saying it right, and Sharon Thompson-Schill put out this paper saying that if you were going to design an experiment and you had the intention to do MVPA, how would you want to design it? Like, would you want long runs or short runs? And they found out that shorter runs actually produce better results. And it didn't necessarily have to do with the size of the training and testing sets as much as the fact that every time you stop and start the scanner, some things will change. And the things that change, I think, tend to be noise related. The subject might move a little bit, or the mean signal scanner might change ever so slightly. And so if you have lots of short scanning runs and you try to find the patterns in your training set that are robust to run, those will be the ones that sort of maximally discount the chance effects of noise. If you just have one long scanning run and there's some confound, like the signals drifting across the run, your classifier might think that that's the important difference between two mental states and miss the critical difference. But it isn't clear from this paper what exactly is contributing to the advantage. That's just a speculation. Okay, so many times when you're creating, oh, sorry, I'll take your question first. Do you want to pass the mic over?
Hello? Okay.
Good.
In this fast event-related design, and if you were to get beta weights for each trial, is that what you're then classifying rather than the pattern of activity? You're trying to classify beta weights?
Yes, yeah, so thank you for clarifying that if others didn't get that. So the purpose of doing any of that kind of modeling is to get these, what I'm calling examples. So to train the classifier, all you need is to have some patterns. The patterns could be brain patterns, they could be fingerprints, weather data, whatever you want. The classifier doesn't care. You're just, in this case, we want patterns of voxel activity. And so we need one for each trial or each mental state. It might not be a trial, it could be a block. It could be a subcomponent of a trial. But we need to say sort of this is an example of one mental state and this other pattern is an example of another one. And so we want to give our classifier some number of these patterns, and those can be derived by taking individual TRs, averages of several TRs, weighted averages of TRs, or single trial beta estimates from a beta series style GLM or the LSS variant of that. So these are all approaches that one could take. Yes?
So I was wondering if you could, if you use this strategy of getting the betas, whether you could use between subject MVPA. So essentially just, if you have 20 subjects, train on 18, test on 2, and you just do all the folds necessary to...
Yeah, so this sort of harks back to an earlier comment I made, and something I do have some slides on much later about classifying across subjects. And this is something that I've done in my data and had some success at doing leave one subject out classification for mental states that are sufficiently consistent across subjects. So if I wanted to, if I was interested in the representation of tables versus desks or something subtle, like maybe you could decode that within an individual subject, but what's the chance that your pattern of ventral temporal cortex activity could differentiate that in the same way that mine does? It seems unlikely, even with fancy tools like hyperalignment, that certain things, whether you're looking at an oriented grading to the left or to the right, those can be very well decoded from early visual cortex in individual subjects. But the voxels along your calcar and sulcus that have certain preferences won't be the same as mine. And so certain things don't do well across subjects, but other things, like whether you're mind wandering or paying attention, whether you're retrieving a memory or viewing something novel, like large-scale patterns, or different, whether you have a rewarding stimulus or a non-rewarding stimulus, or even certain emotions, might have consistent signatures in a broader sense. And so using smooth, spatially normalized data, the patterns do align well across subjects. And hyperalignment is another way to make them align more across subjects, and Haxby, I think, will tell you much more about that. But I think it will allow many more subtle states, like even butterflies versus cats or something, to be decoded across subjects if you do hyperalignment. But maybe not monarch butterflies versus, I don't know, moths or something similar. Different kind of butterfly. Any other questions before I continue on? Oh, another one. Yeah.
So for the resting state analysis, for the connectivity pattern, is it possible to finding cross-subject pattern?
For connectivity-based decoding,
Yeah.
yes, yeah. So connectivity-based decoding is almost always done across subjects. There have only been, a handful of studies that have done this, like, 20 or 30 at this point, that I've seen of connectivity-based decoding. But it usually uses a leave one subject out approach where you have some connectivity signature of a task state, or even it could be resting state. You could try to see, well, can your connectivity pattern tell me whether you have attention deficit disorder or not? And so I just take 50 controls and 50 ADHD subjects, train my classifier on the connectivity patterns, maybe 300 nodes by 300 nodes. So it's just sort of that pattern, whole brain connectivity. And then I have additional people in the classifier. I will sort of guess their diagnosis. And it might, if it's not good within the training set at differentiating the two subject groups or the two mental states, then it's not going to do well in the test set. But if it does well in the training set, you have a good chance of getting this cross-validation. Yes. And I do have some slides coming up about a cross-subject decoding and connectivity-based decoding, although not a lot of content on those.
You also just mentioned that, at least I think I heard you saying something about smoothing. Typically, you don't smooth in MVPA, right? That would be sort of like a big no-no if you start smoothing.
Oh yeah, I wish I had, that's right, I have some stuff later on that sort of deals with the smoothness of the data with respect to decoding visual representations and some controversy about that. But I should have, I've thought more about smoothing since I prepared these slides, and I should have put in some more, because there've been some papers that have come out recently that have shown that a little bit of smoothing is good. So I've generally found for the kind of mental states that I classify, like whether someone is experiencing recollection or familiarity or novelty, that smoothing helps, it doesn't hurt. So the unsmoothed data, the classifier might get like 75%, and the smooth classifier might get 80%. Like it improves the accuracy because these patterns are large-scale patterns. Other kinds of patterns that are more subtle, like decoding different types of male faces versus female faces, like you might not want to smooth, maybe preserving the spatial resolution of the data, or even acquiring the data with higher spatial resolution and then not smoothing, could be beneficial. But the latest paper I've seen that evaluated this showed that even for what we would think are these subtle kind of high spatial resolution distinctions, like that depend on cortical columns for decoding visual orientation, that still about 3 millimeters of smoothing is ideal in empirical data and in simulations. So it just, it improves the signal to noise in a way that is beneficial for the classifiers. That said, in searchlight analyses where people are training classifiers on very small regions that range from like 25 voxels to maybe 400 voxels per searchlight, usually about like 120 voxels per searchlight. Most people don't smooth within the searchlight because you don't want the searchlight to have a uniform pattern. And I will say more about searchlights for those who don't know what I'm talking about as we get to that part. You had a question?
Can you talk about alternative strategies to the leave one out cross-validation approach? Because I know that there was a recent preprint that was deposited, and it shows that there are sometimes pitfalls to this approach. And so what would be some other ones?
Well, the pitfalls can come if there's sort of non-independence, I guess, in the way that you chop up your data. Or maybe you're inflating your classification accuracy relative to if you brought that person back on another day and put them in. by sort of chopping up your data. But I still think if you divide up by runs and have sort of balanced numbers of classes within each run, that it's mostly immune to bias. I'm not as aware of sort of what the major pitfalls of the approach are. Like if somehow your data had a lot of dependence at the order of the trials, it's always like condition A, condition B, condition A, condition B, and then you just leave 2 trials out, like the classifier may be influenced somehow by the structure of your data. Although even in that example that I just gave, I don't quite see like how it would get it right. Because it's not looking at the temporal dependent. It actually doesn't care what the order of your trials are. For a lot of the cross-validation I do, I've switched from using leave one run out to leave some percentage of the data out, and I train my classifier many times, and I just randomly sample from all of my available trials. So maybe I have 400 trials, and I just sample 360 of the trials for training and then 40 for the test, balanced from each class. And then I do it again and again and again with different samplings. And that could be biased if there was like, if the events weren't far enough spaced from each other, and like sometimes when you pick a training set, the testing data might be too close to it and could potentially be influenced. But if you have enough data, those biases are usually not. The classifier is able to overcome like potential biases from the testing trial being non-independent from the training. In most cases, people will leave one run out because at least the run is more independent. You could have not acquired that last run, but you did, and it's like you stopped the scanner and you started it again. The person's brain activity could not have possibly been contaminated by residual bulb from the preceding run. So every sort of data point within that testing run wasn't contributing to anything you trained your classifier on. Whereas if you chop out individual trials, maybe there's a little bit of codependence between trials. Okay. But if you're aware of sort of a particular paper, we could discuss that after. Okay, so feature selection is another important and kind of thorny issue. I think Pamela will touch on this a little bit more in her presentation next week. So selecting which voxels to include in the analysis. In any kind of machine learning analysis, you have many potential features that you could throw out your model. If I want to know if tomorrow is going to be sunny or rainy, like there's many things I can measure about the atmosphere. I can measure the wind speed and the temperature and the pressure and the dew point and all these things. And some of them might be relevant to my prediction and some might not. And the more stuff I put in to the model, I could sort of overwhelm it with noise or irrelevant information and potentially overfit that and get a result that doesn't validate well. So you want to generally try to reduce the features you use. In this case, I'm just going to refer to features as voxels, but they don't have to be voxels. They could be independent components of spatial patterns of activity throughout the brain. Could be your features, so you're reducing the dimensionality from 200,000 voxels to, 20 components and so forth. That's one approach. But if we're doing voxels, we have to do this in an unbiased way. So you can run a univariate GLM as long as you do it within your training set data. So you would identify voxels that have some characteristic that you'd think would make them potentially diagnostic of the classes you're trying to train your classifier to distinguish. And this doesn't, there are different ways of the voxels being potentially informative. One is you could just pick voxels that are task responsive. So voxels in the white matter and the ventricles and other areas, as well as maybe areas that aren't relevant at all to your task, just won't tend to respond very much every time you see a trial. They just won't have a high sort of excursion of activity. And so they might not be informative. You could just pick all conditions versus baseline. You could also pick voxels that have high selectivity. So within the training set, you can do whatever you want. You can make the training set better by picking the voxels within the training set that differ in their mean activity between the 2 conditions or between the 5 conditions, and then train your classifier. So you've biased your training set, but you don't care about that. You just want to see how well it generalizes to the test set. So as long as you leave the test set out, you can try to find information in your training set that will allow you to pick the most diagnostic voxels. So this is just an illustration of, I forget what's being plotted actually in this particular figure, but picking voxels that have high diagnosticity. Okay, so what criteria should define important voxels? Well, I mentioned difference from baseline, so you could pick voxels that have task-related activity, agnostic to whether they differ between conditions. So just as a starting point, we'll throw out voxels that don't have above baseline activity. You could be missing things, maybe the negative going voxels or even the voxels with low signal carry information. So this approach won't always be the best. You could also pick voxels that show the maximum task-related effects or condition-specific effects. So you could just do a one-way ANOVA with however many classes and pick voxels that show some effective task. So task matters in this voxel. It had a high F score, and task didn't really affect the univariate activity of this voxel, so I'm not going to include it. So that's another approach. It's one of the more popular. Again, all within the training set. You can also pick voxels that prefer one class. So I could pick voxels that really like condition A, faces, and then voxels that really like condition B, scenes, and voxels that really like condition C, body parts, and then do a union of those different maths. So I impose some threshold, maybe the top 10% of face voxels in the brain, top 10% of scene, top 10% of body part, and put it together, and now I have voxels that are highly selective for each of the classes within this particular training set, and then I'll train my classifier and see how well it does on the test set when tested only on those same voxels. You can also pick, I don't see this done as much, but it's kind of a neat approach, just pick voxels that have high stability across runs. So you don't care what the voxel's pattern is, if it tends to respond to condition C a little more than B, a little more than A, as long as it shows the same pattern of condition preferences across runs. So you sort of look at its similarity of its condition preferences across runs and say, well, any voxel that's stable in each of the runs in this sort of these condition-specific estimates is going to be a good voxel. And voxels that vary a lot across runs, so next run it prefers condition C, that's not going to be good for my testing data. I want voxels that show a lot of consistency in their preferences, regardless of how strong those preferences are. Okay. So another way that sort of avoids any potential pitfalls of what we call peaking, so you want, as long as you're doing your voxel selection within the training set, you should be okay with your testing set. But if you're concerned about that or want to do it in kind of an easier approach, you could just use independently defined ROIs. These could be based on anatomy, like from the AAL Atlas or the Harvard-Oxford Atlas. You can pick individual regions that you're interested in. You could just put them all together and say, well, I'm interested in all the cortical areas, but I don't care about the ventricles or the cerebellum or the white matter. You might not care about the motor cortex, so you could throw that out, and so forth. And then you can even, if you have a set of ROIs, you could just train and test a classifier on each ROI, and then put the data back in the brain, and maybe even turn it into a nice 3D plot. So this is just looking at classification accuracy within each ROI. This region did the best on this particular contrast. Regions in darker colors did more poorly. Regions that don't appear didn't do well at all. So you sort of get a sense of where in the brain there's significant information about your classes. And this is, in some ways, kind of like a poor man's searchlight analysis, that you're, instead of doing 200,000 searchlight spheres at every possible location in the brain, here you're just training it on 90 ROIs, and you get 90 classification accuracy values that you can plot. And these maps can sometimes be useful. Okay. Sometimes getting rid of uninformative voxels using feature selection doesn't actually help very much. So this was an example that from my own data, classifying memory states, and no matter sort of how hard I tried to push on this sort of ANOVA-based feature selection within the training set, the classifier never got better. I never found sort of a sweet spot that with 800 voxels it was doing awesome, and then when I included more voxels it started to do more poorly. As I added more voxels, I got diminishing returns. So with the top one voxel in the brain from the training set, and I used that one voxel's activity in the test set and tried to tell if the subject's seeing an old face or a new face. It's including here, the classifier accuracy was like still about 70%. Or I don't know what these you know, probably like 67%. And with the top 25 voxels, it did better. And you can see that as I rise up here to about 2,000 voxels, I just keep getting better and better, that there's more information. And then as I start doubling, each one of these is a doubling, I pick the top 3,200. it's getting better, but not that much better. And it sort of plateaus out here to the mask here was 23,000 voxels was the whole mask, and that was just ever so slightly better than 12,800 voxels. Now, other people have seen this as well. So this is a paper that came out a few years ago that did this for a bunch of different kinds of situations and regions, classifying Alzheimer's disease versus normal controls, MCI versus normal controls, and so forth. And they just found that the number of features that they give their classifier, what they're calling here sample size, that accuracy levels saturated, and that they didn't see any benefit. Well, here sample size might actually be the number of subjects, but in any case, that the feature selection, trying to improve their training set by getting the most diagnostic information here, did not help them. Other studies find sort of this sweet spot. So this paper found a sweet spot around 1,000 voxels, and then performance kind of drifted off as you added more. This paper found a sweet spot that was fewer voxels. And it depends on what you're trying to decode, whether it's sort of memory representations or attentional states or visual stimuli. You might find a different sweet spot. And you can sort of search for this in your data, but the more you search for it, the more you're potentially biasing your results. So if you use every possible feature selection method and then just pick the one that was best, in essence, you did look at your test data. You looked to see which choice of parameters maximize the accuracy at which I could decode my held out data. And so the chance that you would do as well on a new data set is quite low. You've biased it. It's hard to get around because you don't know at the beginning all of the right parameters, so the right pre-processing steps, the right way of defining your examples, the right way of chopping up your data into training and testing sets, and the right way of doing feature selection. These are all things that you can play around with and try to improve your results. But the most unbiased way to do it is to do it. So let's say if you had 10 runs, you could leave one run out as your testing data, take 9 runs as your training data, and then to subdivide that. So now 8 of those runs will be my sort of nested training set. I'll leave that 9th run to be the test set. I'll try to maximize performance on that. And once I pick that set of parameters, then I can apply it to the data from the patterns from the 10th run. And so you end up with different parameters each time because you sort of pick parameters that maximize the results within the training set, holding a piece of the training set out to determine which parameter settings were best within your training set. And then you use those same parameters, in this case maybe the level of feature selection for your test set. And so I've tried that. It's very computationally intensive, and it also makes it hard to write the methods section because you have different parameters for each fold of your cross-validation. But it's much less biased than just trying everything under the sun and trying to get your classification accuracy as high as possible. Okay. So, okay, I'll continue here. So this is a figure that's kind of similar conceptually to some of the illustrations I've shown. It's just giving you sort of a sense of how the classification process works. So once you have a series of trials, let's say you have 100 trials, 50 of each class, you can subdivide it up. So you can label them. Well, the usual way that we do this in a univariate analysis is we just label them as being from one condition or another, and we do a statistical contrast, and we get these kind of SPM-style maps. But instead, we could divide some of the patterns up and make sort of a set of training patterns and then leave some out. These are our test patterns, and you feed them in to this multivariate classification algorithm. And based on how much of a match in a multivariate sense this pattern is to the weights that the classifier has learned, the weights that differentiate condition A examples from condition B examples, the classifier will make a prediction which can be turned into a probability. 65% likely that this test pattern is from condition A, and 35% likely from condition B. And as I mentioned before, I find these probability estimates to be very interesting. So most people just force their classifier to guess, and if it said 51% condition A, you guess condition A. If it said 99% condition A, you guess condition A, but there's a big difference between those. In one case, the classifier is viewing this as very paradigmatic of its sort of multivariate representation of what condition A brain patterns should be like. In the other case, the classifier is basically trying to tell you, I don't know, but if you really force me to guess, like maybe I would say kind of condition A. And those guesses are very different. And so you can, if you were interested in how well the classifier would do when it was really confident, you could sort of give the classifier a third option that if the probability is greater than 80%, guess that class, or if it's between sort of 80% and 50%, say, I don't know, and then your classification accuracy will go way up at the expense of having many trials where it says, I do not know. But in practical situations where you don't want to make false positives, if you're trying to diagnose a disease or diagnose some subject's psychiatric condition or trying to tell if someone's telling the truth or lying or not, looking at these probabilities is useful. Maybe this is even the measure you want to report. How much did the classifier, how much evidence did it think that this was a truth-telling event versus a lying event? Or did your algorithm basically say, I don't know? Okay, so maybe, I don't know if we should take a break. I have more content to present, but I'm kind of up against, we were gonna do more truths and lies, right? I might skip some of the stuff about the classification models and just I'll go up to noon and do a few practical things, and then from 12 to 12:30 we'll do more student introductions and start lunch at 12:30. So if you don't mind, I'll continue. But after lunch, okay. But so we're going to do lunch from what time? Are we scheduled 12:30? Oh, okay. Well, I'll keep going until for at least the next 10 minutes. Okay. So these were some slides from Francisco Pereira. Okay. About classification approaches. And so if you had two voxel, any classifier sort of representation is really only easily visualized if you have two features, two voxels here. Otherwise, it becomes multidimensional and it's hard to see. But if you've, in these toy examples, you just have two voxels that are for two different classes, tools and buildings. And the simplest function to determine sort of which example is this is an example of class A tools or class B buildings. It's just nearest neighbor, sort of is it closer to these examples than these? So that requires some similarity metric. It's nearest in what sense? Well, it could be Euclidean distance to sort of the mean of this class and the mean of this class for each voxel, or Euclidean distance and sort of the multidimensional scaling representation. It could be just this correlation of this pattern to the mean pattern of these voxels. That was the original Haxby approach, and it's still sort of the workhorse of RSA-based analyses. But this is a very simplistic kind of classifier model. You could do something a little more complex, like train a linear discriminant, and there are potential separating boundaries that could separate these classes perfectly in this toy example. How do you know which one's the best linear discriminant A versus linear discriminant B? And you want the one that sort of maximizes the ratio between you want really low within class variance and really high sort of across class variance. And so you want to maximize the ratio of the variance between the classes to the ratio of the variance, to the variance within the classes. So that's the LDA, linear discriminant analysis approach to find the separating boundary. And that was used for a while, but it's, I think, less popular now. More classifiers now tend to use things like regularized logistic regression. We've used that quite a lot in my own work, as well as linear support vector machines, which we use as well. And that's probably the dominant classification model that people are using. Gaussian naive Bayes classifiers are very fast to estimate, and so they work very well for searchlight analyses where you're running 200,000 classifiers in each person's brain for each sort of pair of conditions of interest. So you need to do this many times, and sometimes a support vector machine, if it's taking 50 milliseconds per searchlight, and the Gaussian naive Bayes, if it's taking 1 millisecond per searchlight, it might be more efficient to use a simpler algorithm. I'm not going to talk about the mathematics of these, but I'll show just a few examples of what you can extract. So these weight matrices that any classifier will produce are interesting, but potentially subject to misinterpretation. So for any voxel, you can see, is it weighted sort of more toward, if you had two classes, sort of it gets a weight for class A and a weight for class B. Often that's just the additive inverse of its weight for class A. So the voxel has a weight of how predictive it is. And every voxel has a weight, and basically the sum of the weights times the activity of a given pattern can give you some number of how likely that pattern is to be an example of class A or class B. There are other functions for sort of taking the trained classification weights and making the prediction, but essentially it's akin to taking the feature weights at each voxel times the patterns of the observed data and trying to see where it falls on that boundary. So the weights will give you some sense of which voxels the classifier found to be particularly diagnostic for one class or another. But the weights can't sort of tell the whole story because the classifier has this rich multivariate data set, and any individual voxel, even if it has a high weight, might not be that critical. You could take out those voxels and potentially do just as well if there's redundant information elsewhere. But it can be useful to look at them to sort of see which areas are being leaned on heavily. Now with linear support vector machines, just like the other example, there are many separating boundaries here. These are two classes that are well distinguished. How do you pick sort of which separating line your classifier is going to learn? In two-dimensional space, it's just a line through this plane, but in multi-dimensional space, it's kind of a hyperplane through space. And the classifier in SVM is just being told, well, pick the one that sort of maximizes the margin between these two planes here. So you have sort of one margin here that's like the endpoint of this class, we'll call it class A, and then one that's sort of the endpoint of examples of these classes as they get closer together. So you want to find this line that when you sort of expand from the line in orthogonal directions, sort of these parallel directions, will start to hit data points in a way that maximizes the width of this margin. And these points are what is called the support vectors. They determine these sort of separating planes. And the further that you can separate the margin between the two planes, this plane for class A and the plane for class B, the wider this margin will be. And there's going to be one solution for any sort of class of the separating plane here. In this case, just a line, but you'll see in another slide, it's really sort of a plane through multidimensional space that separates these two classes in an optimal way. And the SVM solution is a very robust solution for determining the just optimal decision boundary between two classes. Okay, so you're finding the linear decision boundary that maximizes this margin. So sometimes classes are separable, like in these examples, with a linear function. And sometimes they're not linearly separable. Even with two voxels, there could be some nonlinear relationship that when voxel activity 2 is high and 1 is medium, you get a different result than when 1 is high and when 2 is high and 1 is high. And in that case, you might need a nonlinear classifier to distinguish between these classes. So nonlinear classification boundaries can be represented as linear boundaries on some transformed or projected feature space. And to illustrate this, I like this slide that sort of shows that, well, if you have two classes of examples in one dimension here, so class A has values that are kind of negative and then some values that are positive, and class B has examples that are intermediate, and it seems kind of hopeless to create any line. If I put it here, I'll get 4 of these right and 3 of these wrong, but I'll get all of these right, and if I put it here, I'll get some of these wrong. There's no great place to put a single boundary in this one dimension. But if I were to take just the square of this, so instead of x, plot x squared, now I can create a separating boundary that separates these classes perfectly. So this is just a quadratic function. It required a nonlinear projection of the data that then allowed me to get perfect separability. And this can be expanded to multiple dimensions. It's when you do an SVM analysis, you have to specify sort of what kind of kernel you want to use. People, when they first started doing this, were using artificial neural network models for classification because those were sort of dominant in computational psychology approaches and seemed well suited for neuroimaging data, where you would have each of the voxels feeding onto some number of hidden layers, and then you would have an outcome matrix, and you could just sort of train the neural network model, and it would learn some weights between the voxels and this sort of intermediate representation, a latent representation that comes from these hidden layers, which then project onto your different class or decision outcomes. And this approach did it reasonably well, but I think it's fallen out of favor because approaches like support vector machines or other simpler classifiers do just as well, if not better, and provide more interpretable measures of what the classifier has learned. Looking at these hidden layer weights is very tricky because it imposes this nonlinearity. And so with support vector machines, you have a more intuitive mapping between voxel space and your outcome measure. With a quadratic SVM, like this example, it's a little bit trickier to understand what the classifier has learned than a linear SVM, but it's going to be more easy to interrogate than a neural network usually. And nonlinear classifiers, one should be cautious about. You often have very few examples in fMRI data and many features. So you have many voxels and few trials. And so these models are prone to sort of overfitting. There's some optimal trade-off between, in terms of how well your classifier does. You want to get low prediction error. And you can increase the complexity of the model by adding all these nonlinearities that lets your classifier sort of carve up some space that separates your data. And this drawing is meant to say, well, you could just draw sort of some swirly thing around all the examples of your data within the training set in multidimensional space, and then you're going to do really well on your training set, but it won't generalize well to the test set. So having a model that's less complex, like a linear classifier, even if it might not do as well in your training set, could actually be better at generalizing to your testing set. So I will just stop here by saying that in the field, if you just plot the literature up to, starting from Haxby's first paper to 2011 when this analysis was concluded here, linear classifiers have been dominant in terms of the number of studies, and nonlinear classifiers have never really caught on. And I think it's because of overfitting problems and also the complexity of interpreting the results, and the fact that they just often don't outperform. You can try different kinds of nonlinear SVMs, and you won't necessarily get results that are any better than your linear SVM. So most people are still doing linear classifiers, and in some ways the field has even gone further, and many people are just going back to the original sort of Haxby 2001 representational similarity analysis. They're not even classifying anymore. They're just saying, well, is activity in this condition more similar to condition A or condition B? This just spatial pattern of bold activity looking at the Pearson R correlation. And that has, if you were to graph that, I mean, that's sort of rose up here, dropped off for a while, and it's been coming back because Nico Kriegescourt really revived it in a major way and showcased that there's extreme power in that approach for doing all kinds of interesting things. And it's less stringent in some ways than these classifiers. There's fewer assumptions. Okay, so I will, I'll stop there and we can take lunch. You have all of my slides. I'll look at the schedule. If there's time, at some point I can continue on with this because I have more content than I was able to convey now. But if not, you have these slides, and I'm happy to talk with you about it. I'll be around for the next 2 days. So thanks.